I am happy to participate in this symposium and serve as a coordinator. And let's introduce the last speaker, Dr. Zhao Shu-Miaw, Distinguished Research Fellow at the Biodiversity Research Center of Academia Sinica, where she studies the comparative phylogenomics of seed plants and inferred from plastid, mitochondrial, and nuclear genomes. So without further ado, let's pass the time to Professor, to Dr. Zhao.
Thank you, Professor Huang, for the introduction. Can you guys hear me clearly? Yes, yes. All right. I would first like to thank SSE for inviting me and give me the opportunity to share with all Chinese people from East Asia with my recent research results. So this is project manager Wu Zhongxian and also Chen Zhongyi from NPU. So we work in collaboration on this research. We want to look at the plastid phylogenomics and plastome evolution in the morning glory family. Some people translate into transome, some people have different system, but systematic and phylogeny in Taiwan, we have a different translation. But I think in China, you translate it into a different one. But in any case, that's what I meant. So there are two topics of my subject. First is about the label structure of plastome evolution. If you want to sample a majority, then you will identify some mutation, and this mutation would become a very important markers. We also find that within the mitochondrial, I don't want to use there's actually mitochondrial DNA. So this is actually very conversive, and they do not like to accept other people's DNA. So if you want to identify that there's mitochondria, then you can actually find. So we are actually the fourth projects that find this. So we also identified that there is actually a different case. That's the prosgenome. I think I will make it clearer. Yeah, there's actually a loss of plastic intron there. But the loss of this intron may not be as simple as we seem. It may not because of the cDNA and the loss of transcription during the process, and just say, oh, the other cDNA would just then insert. So we find that it's not as simple. That's why we proposed a new model as a hypothesis to explain why is that the plastid intron would be lost. The other thing that we also talk about is the phylogenomics. The thing is that you have to have enough, coverage. That's why that if you look at previous literatures, you will see that where the conflicts and conversationalities are, and then you look at it. Normally, it's actually because the sampling are not broad enough so that you don't see enough variations, and that's why the genomics they propose may not be standard. Let's firstly quickly define some terms. First is plastic versus chloroplasts. Initially, when we're doing chloroplast, we need to actually extract all the chloroplast, and it takes a lot of time. And within one cell, you have around 50 chromoplasts. So among all the chromoplasts, they have around 20 to 50 genes, genome. So within one cell, you have about 2,000 to 3,000 genome. In the past, if you want to extract that, of course, it's not easy. It's hard to get something pure. But starting from 10 years ago, we started to have NGS. So we extract it from flowers, roots, or the leaves, so you can do the whole tissue. So we no longer call it chloroplast. If you want to do the full PNA, you have to call it plastid. Plastid basically has its biosynthesis, which is chloroplast and also chromoplast. On the other hand, in terms of preserving, that is storage. This is, for instance, sweet potatoes. They have a lot of this storage area. So This is why we call it plastid. And this research, we will use the word mutation. Mutation is actually referred to variations within a population. If it's actually seen in a generational change, then we call it substitution. That means that there's speciation, and we use the word substitution to identify these differences. So we will use mutation and substitution quite frequently, but they refer to different things. This is just to clarify. And vertical and horizontal or lateral transfer. So the vertical is that the transfer of genetic information from parents or offspring, which means that there's no problem in reproduction. As for the horizontal transfer, it means that it is actually across different individuals and different species. This is actually a mating across normal mating barriers. So the passing of the genes is actually not from parents to offspring, but more like the sympacian have this species, whatever reasons, they sort of like integrate together. So it's easy for some plants to have this horizontal transfer phenomenon. And in the past, we look at it based on like phylogenetics and phylogenomics to actually restore the past history. So we may call it phylogenetics or phylogenomics. Now, in evolution, we normally call it evolution. We have a different translation for evolution in Taiwan. We call it 演化, not 进化. We're not calling it a progression. We are calling it in a more neutral term called evolution. So this is the basic structure of the plastic genomes. In the past, we think that this is actually a closed circle, just like a mitochondria. But for mitochondria, it's not necessarily a circle shape, right? Circular shape, especially especially in plants. We all know that these gradually comes from the original cells. In the past, it has like thousands of genes, and some gradually goes to the core. So basically, this is actually if actually has some more complex structures, it can be aggregated into somewhat like a jellyfish shape. So there are a lot of lines over there. So this is an IR and SSC and LSC, and LSC and SSC also plus a pair of inverted IR. So these are the general structure of this. Basically, they have around 100 genes. This SSC over here is quite conservative, and it's very slower in changing, and these IRs are very important. It's easier to make changes and variations, and these two, it's doubles. So amount, I would say 100, but actually they only have about like 80 single copied genes. But you are able to form it into a circle, then you mean that means that you actually captured almost the majority of the genes. So when we're doing the sequencing with 20 giga, you can actually get a quite comprehensive genomes. This is a very typical example. Let's have a look. We actually put the LSC over here. These are the junctions. On the right-hand and left-hand side is IRA. So on the right-hand side is IRA, left-hand side IRB. Just remember the order. And this is a gymnosperm and angiosperm. Over here, you can see that for the LSC gene, it continues to go like this, and the YCF2 will go to over there. So this here will be doubled because of IR. And then angiospots, let's have a look at. There are fewer genes here, and the LSC goes to IR. So IR gets more genes. So this is the initial sample that we made in 2007. Then in 2016, the Kunming Botanica Institute actually did a lot of research and found similar findings, but it shows more of the changes roots. You can see that the IR get longer and wider. And this is quite clear, right? Getting wider and everything. And the gene over here, you know, the other side would get more conservative, but you can get a good marker in this. So today I want to focus about the in Taiwan we call it Convolvulaceae. Well, we call it a glory morning and Qian Hua in Taiwan. So the reason we call it is that because they are normally in the ground and they would actually combined and together. So and when there are cows passing by, they would actually trap the food of the cows. I actually made a song for this flower. I will sing that song to you if I have the opportunity. But anyways, I just want to share with you that this is what we are talking about. The convolvulations actually look at this, and there are 60 different genres in this, and there are 2,000 species. So it's a large species, and it's very hard to give it a more thorough studies. So there are actually something that we're more familiar with. And since Convolvulaceae, they have different colors in its blossoms, just like the first slide that I share with you. It's very beautiful, very different slides. I will show you the picture later. So Japanese people, when morning glory is like the face you have in the morning, so that's why they call the Convolvulaceae morning glory. But in the early studies, you can see that it was divided into 9 tribes. And these two studies are only 5 years apart, but they are quite different. Let's have a look. This is put in a different tribe. See? Can I remove this because it blocks me? Oh, okay, I can move this. All right, no problem. Then the recent study looks at the impermonia. They use a 353 kit, and with that you can actually create a lot of different analysis. And this is very different than mitochondria. As you can see, these red ones comes over here, whereas the plastid CDs as there are 2 differences. So that means our lab happens to be doing this. We're looking at the genome of water spinach, and we're comparing it with a wild species, and we find that it's quite problematic. And since we need to, it's difficult to collect all 9 tribes. So we ask Professor Chen from Pindong University to help with us because they know where they can find these species in southern Taiwan. But in the past, they were saying that is it one species? Is it within one family or not? So we are doing this exactly. So we want to re-examine the intertribal and intergeneric relations with Convolvulaceae with more comprehensive taxon assembly. And we also want to estimate uncomplained plastic substitution rates across Convolvulaceae because we feel like the substitution rate is quite large, and I think that will make our study more comprehensive. We also want to elucidate the Plastomic variations across Convolvulaceae. And we really identified 2 very important findings, and they're quite interesting, and we can make them into a very good report. So these are some of the materials and methods. We have 75 plastomes across 17 genera, including 19 newly sequenced ones. A lot of sequencing are now there. You don't need to make a lot in order to get them. So this is a sequence platform. You can see plastome. You can see that we get 20 giga for each species. These are just the routine method. This is more important, and in terms of comparison, I will elaborate that part later. So I will share with you the results. I just want to share with you, these are the different colors of this morning glory, and the leaves also look very differently, but they're so beautiful. Every time I wake up at 5 o'clock in order to do the assembly, because a lot of times in the afternoon and the evening, the flower was actually closed, and it's hard to know what color of these blossoms are. On the left-hand side is the evolution tree that we found. Let's have a look at the cascada. The cascada is actually included in this whole Convolvulaceae, and this is only all in one group. This is whereas the Solanaceae is an outgroup. And over here, it's shortened to a 1 in 10, 1/10 because the thicker ones are longer. Otherwise, I would not be able to fit it into one picture. But This is not just a parasital plant, and you can see that this is shorter. That means that it's slower in evolution. This Meramenia, I know it's very difficult to pronounce all these, but this is not meramidic, and it's different from this one. So you can see this is similar to this this study that they do, but you can see that the Cascadia is also not, it's not an independent family, and our results shows a consistent result as theirs. And that's compared the DS and DN rates. We want to look into what, you know, the sequencing, the chronology of it. So we put the years over there so you would know that what's the evolution rate. So we identify 3 points as the timeline, and we see that what would be the substitution rate. So the relative substitution rate and the absolute substitution rate. Let's have a look at the blue one. This is over here. You can see this is quite slow in evolution, and but for Cascadia, for the Cascada, these are more parasital plants, and for these plants, they don't need to work, right? They just eat and survive on other people. So why is it that if they don't need that, and they don't their crawling plants do not need to work, why is it they're still slow in evolution? And all these others are very fast in evolutions. So these are slower genes, and these are faster evolution genes. So let's think about it. Does that have to do with IR? So if this has to do with IR, let's have a look at the left-hand side. Let's look at LSC and SSC, and we put it into a straight line, and then we list out all the IRs. And you can see the gray area, there are some of the genes that are already absent. The one marked in question mark is uncertain. The truncated is in light blue, whereas present is in dark blue. So the slowly evolving clade is over here. These are more, the genes are inflowing, whereas the rapidly evolving clade, the IR will change more significantly. And you can also identify over here that the IR loss, if you look at it from, these are shared among families, 2 different families, whereas the RPL2 intron, the loss of this intron is actually seen in all the convolvulations. So that is something that is a normal character involved afterwards. So that can be a very good marker. We also identify something interesting. Let's have a look over here. If you're only doing phylogeny, then it's hard, right? It's hard to be accepted. But we find something additional. We found that the PSDA is normally close to IRD, but see over here, there's actually a EKB insertion. So we want to enhance it and find that these are mostly, even for Embrya, they also have that. Embrya is actually the most original ones in Emerspan. So that's why we want to know if we assemble it correctly. So we want to verify if this foreign DNA is actually because we assemble it wrong. So there are several ways. We try to see if there is any difference with orthologs. So we use different assembly tools. Originally, we only use once, but then we add a second tool. But you find that the results of assembly is the same. Then we try to check the sequencing coverage. Are they the same or not? They would definitely not have the enough coverage if we make something wrong. And we also check it with PCR assay. And we find that this is how it's enlarged. And these different colors shows the potential origins. And we found that, yes, this is a very diversified definition, and we separate it into 2 parts. And we find that, yes, it's 9K. So that means that we didn't assemble these segments wrong. There are these differences, but what exactly they are, where did they come from? So we do the blast search, just like what I shared. And for mitochondria, there's also it would be higher ones. And yes, indeed, it's very high. And then unfortunately, since it's non-coding, so we cannot transfer it into a cytogenetic tree. We know that this is angiosperm mitochondria DNA, and we know that this is actually mitochondria from angiosperm to the others, and this is also a big story behind it. We just had an article that just got we just submitted another article, because from another plastid to come here, it has to go through mitochondria for the mitochondria to carry it. So let's say it's species A wants to go to the morning glory, then mitochondria plastid needs to first go to the mitochondria, and the mitochondria will then carry this to the mitochondria of the morning glory, and then they will carry it to the plastid. So this is something that we think would have a higher possibility. And another interesting issue is that we found that it's rapidly evolving genes. So we want to know exactly what happened. And we found that for dichondra, the RPL16 as over here, you see that the typical RPL should be over here, but over here they have an extra pair in each IR. So we try to analyze it and find that this is the typical RPL16, and yet over here there is did I calculate it right? Yes, there is like it is exon 1 and exon 2, and this is IR over here on the left hand side. This looks like this from here all the way to over here. These are the conserved, the high identifiable area. But why is it there is actually a blue ones that are repeated? So the other IR would be more special. There's no intron. I didn't write it out here, but there's no intron here. The typical RPL16 can be formed into this one. There are 5 domains over here. And this domain is actually in the spiceromes because it needs to be combined with the spicerosome in order to flex. So that's why we're asking, in IR, is it still active? It's still active splicing? And second question is that we say that they have 2 repeats, and this repeat occurred only lead to like the DNA recombination, and it will become a concern. Would they actually means that they will actually lost the intron because of that? So only one, either 1 or 2 would stand. So we want to check which one stands. So we find that there's an absence in the RPL16 IR copy. All these are lost, and also because on the right hand side is also lost, right? So that's what we're checking. So we had this hypothesis. So we proposed the following. We are saying that this RPL16, it is these 2. These are repeat, direct repeat. And over here, there is an additional intron. Is it because this is then developed and further into this one? So because of some reason, it is then into the IR, and then it's lost, and then it has the further development because IR, we all feel that it has a stabilized function. It's not yet completely confirmed yet, but I think in a replication, it would be, that's where they would do a recombination. All right. Let's compare this. This is exon 1. It is quite small, and this is where the intron is. It has this, but for this one, I have this star mark over there. There's something lost. And the IRD over here, see, there's a missing of intron. And for RNA, we also does RNA analysis. And we do the transcription, and we found that over here, they all have this. Now, let's have another look. This is our model, intron loss model. Intron loss model. So we try to also do a DNA mapping. If we're doing this combination, then this one is the one that the intron is lost. And this is our RA mapping. This is quite typical because at first it's small because these 2 are IR, so it would be it should be double the amount. So that also matched. So the matched DNA sequencing reads is more. Then let's have a look at PCR assay and have a look and see if they have the same amount. So we put it over here. There's an IR. So you can see that they have very similar amounts. And since they have two IRs, one for each side, so it's double the amount. However, if we look at RPL16, and you can see that for RNA, for RNA16, if we look, it's still mainly the LSC major. So over here, the major is quite large. For minors, it's only this, even for these two. So the PCR, we try to look at it from different cycles. We use real-time PCR and treat it with real-time PCR. So our conclusion is that for phylogenomics, we reconfirm the monopoly of Convolvulaceae, including Cuscuta. And we also highlight the extensive variation in substitution rates. And we think that that might relate to the uncertainty of the intertribal and intergenetic relations in Convolvulaceae. In the past, we found that it's different, but it's actually because there's just too vast variance in substitution rates. So you can't compare it with the substitution rate. You still use to see its unit time, and you have to look at the structural change. And as for plastomic evolution, our contributions are that we tell people that this paper illustrate the labioplastom due to multiple rounds of rearrangements and IR contraction expansion during the evolution of Convolvulaceae. And we also find a rare event of mitochondrial DNA integrations in angiosperm plastoms. That's quite rare. We even recently we find genospan, and that's even rarer. We also proposed a new model to interpret loss of plastic intron. It doesn't need to necessarily go through a cDNA. It can go through the repeat mediated homologous recombination. But you will feel that it comes to IR and then has an additional copy. What's the function? We don't know. But with an additional compound, why is it the NGN span would actually compete and will actually win out? The intron is actually quite short, and that means that the gene is actually quite intensified, and that means that things are coming very faster. It's just like a production line. They have like 5 rows of people doing this. You do not need to go through anything else, and then it's faster. And since they have more genes, because they are all go into IR would double the amount, so it would make things more efficient. Our paper was published last year in Frontier and Science. So if you have any questions, I think you can all have a look, and any suggestions are welcome. I would especially like to thank the National Science and Technology Council in Taiwan, and also the funds from our Biodiversity Research Center. We have this very cute this very cute picture, and it's like a heart-shaped leaf. I also like to especially thank SSC for giving me this opportunity, and thank you for your participation. Thank you for listening to my presentation. Thank you. Thank you, researcher and Zhao Shuliang, for this wonderful presentation. I actually learned a lot, because in the past I've never looked into the exact progress of chronoplasts. But just like to open the floor for any questions. You can either ask a question in the Q&A or in the chat box. All right, then I will just start with the first question then. Yeah, yes, sure. You mentioned the evolution rate of chronoplasts is actually very fast. Do you have some hypothesis? Whose substitution rate? You identify like some of it, right? Yes. Any hypothesis you propose to explain this phenomenon or implant? Why is it that the substitution rate is higher, and what's the relevance? Yeah, I actually show you a graph. This is over here. That's we should call it plastid. It's not a chloroplasts. So I'll stick with the term plastids. So this is what happened. This is the earlier evolution. This is the later evolution. And you can see that this is 10 times fold. And it has to do with its environment. And if the substitution rate is higher, it's normally because there's loss of genes or rearrangement of genes. And it must be because of some changes or situations happening in the environments. And the genes are not just lost, it's transferred. No, mostly are transferred to the others. And also it may be more efficient with this transfer. So it's not really lost. Let me give you an example. Let's say we see these some plants, they do no longer have these chloroplasts. But why is it that the custody you have this? I think so. It's not different. So I think we want to know the genome span. That's is the conifers and everything. Conifers, it's not a monobiotic, but it's it's actually so one of us has this A, but it's IRA, but the remaining they are there. They have B, but it's still there. It's still thriving. So I think we done this like more than a decade ago, but I found that even though there's loss of this gene, but they will actually insert a repeat. So you can see that there's all these repeats in it, right? So that's a Giacomodia, and you have the repeat, and it would actually make this stabilize. They has this Is and Ir, and it wants if we want to do duplications, and it becomes this IR and everything. So they would actually become like a scar, and the repeat actually comes from the IR. So it start with the IR for the repeat. You will have to start with one strand to two strand. It may be related to it, but at this moment, this is a big question. But as long as you start with this evolutionary tree, and you find that, whoa, that's a very long tree, and there must be there is a structural trend in between, and you will definitely find something interesting to discuss, and you will definitely be able to publish another article. That's a big question. That's not a small question.
我想问一下，那个是指，那个吐司指是不是不在现在不在选化科里了，还是怎么？
My question is that the Cuscute, is it a change into a different genus? Yes, it is now Cuscute because it has longer branches. Yes. It's you can see it doesn't have a long branch. You can see the difference on the slide. So it's not necessarily longer. From our observation, more than a decade ago, we didn't have NDS at that time. So I realized that there are greens. They're not only just palely yellow. It actually is photogenic, and we were thinking about we should look at the samples of different stages. So that was an interesting topic for me, but I didn't know how long it would take to do a transcriptome. So it was about almost 20 years ago. So we dropped it. So when the seeds sprout, it would be green. It's not pale. The embryo was green. Yes. You can buy seeds and try to grow the plant yourself, and you'll see the color change. Oh, I forgot to mention one thing. Allow me to add. Henry told me to talk about insights or field research. My recommendation for everyone is that you need to look at what people are doing, especially when you want to submit a significant lab or level. If you compare their work and they're different, and you can just contact the lab and you can work with them, because you guys can have new discoveries, comparing contrasting your previous work. Just take this graph as an example from the different species phylogenomics, you can there's so many things that you can dig into, and we can compare all the different branches. And a lot of these are stated in the papers. And as you've noticed something, you can dig in. So for our paper, we just discussed two things. There is a difference in mitochondrial sequence with a plant of a lot of differentiation and varieties. And the second topic would be the repeats. It was really interesting. We worked on Pinancy and carnivores, and we noticed a lot of things like this. So we published so many papers on this. So this is what I wanted to add. Thank you. Okay. In the chat room, there are two um, speakers saying that they want to hear your song. They want to hear your song because in your presentation you said that you wrote a song for the flower. Oh, it was a song of Morning Glory song. I can record it later, sometime later, and I'll upload it on YouTube because I really can't sing. So I only dare to sing for myself or sing to my family members. When I retire, when I have time, I'll record a song I wrote about Morning Glory. Thank you. Okay. I think people are high. People are too shy, or perhaps I did too good or not good enough so that you don't have enough questions. But nevertheless, thank you for this opportunity. Thank you for making time to listen to me. Heard so many interesting lectures today, but I don't really I don't comprehend it to 100%, but they are very interesting. Aren't we time isn't the time up yet? Thank you, Dr. Zhao, and thank you to Dr. Huang for moderating. So let's move on to the next panel. So feel free to jump in because it's a free discussion at this point.